Good presentation. Thank you
Thank
again.
you. Mhm.
Hello, and thanks for joining us today for the latest Technology Networks High Throughput Screening webinar. My name is George Hammond, and I'm a conference producer for Technology Networks, and I'm excited to be here to moderate the event today. I'm also very pleased to introduce Dr. Chun Wu presenting his talk, A High Throughput Virtual Screening Approach to Identifying Novel SARS-CoV-2 Inhibitors. Dr. Chun Wu is a tenured associate professor with a joint appointment in the departments of chemistry and biochemistry and the biological biomedical sciences at Rowan University. At Rowan, using genomic datasets, his lab is developing a novel evolution theory, not only to best explain the molecular evolution of SARS-CoV-2 and other pathogens, but also to accurately identify hotspots in their genome for developing next-generation vaccines and drugs to treat these infectious diseases. In Dr. Wu's talk today, he will present a case study for the discovery of 4 non-nucleoside small molecules that bind favorably to SARS-CoV-2 RDRP by utilizing a high-throughput virtual screening against the vast zinc compound database coupled with extensive molecular dynamic simulations. A warm welcome to you, Dr. Wu. Following the presentation, we will have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. Please also feel free to navigate over to our literature section where you can download the latest resources from the sponsors of today's webinar. If you experience any technical difficulties throughout the webinar, please click the chat icon in the bottom right corner to request support. And without further ado, I will now hand over to Dr. Wu to begin his presentation.
Okay, so good morning, good afternoon, good evening. Okay, and it's a great honor to present our research and thank George for this nice introduction. Okay, so let's go straight to this talk. And okay, here's the outline of this talk. So in the first part, I will introduce you some of the basic computational method for doing the drug discovery. And then I will move to the specific project we will discuss, which are kind of develop a novel inhibitor for the SARS-CoV-2 RNA-dependent polymerase. Okay, so that's our plan. So let's start our first part. So first thing, why we want to do the computer-aided drug design? Okay, the reason is very simple. The traditional way is just took too much money, too long to develop a new drug. So like pandemic this time, we cannot wait for years, and we even cannot wait for months sometimes to save the life. Okay, and why the traditional way cost so much time and money? Because the way it does is the blind testing approach, and they just draw the lottery, try 10,000 compounds, move through the pipeline, do the experiment, and then hopefully there's some the active one. And then so you're spending so much time and resources on bad compounds. Okay, so we use a computer, we can kind of eliminate those bad compounds by doing the virtual screening, use the information of the drug target. We know it's a sequence, we know it's a structure, we know a lot of the information about our drug target. So in the computer, we can set up the virtual screening. And then we use computing power to screening millions, billions compound, even compound has not been synthesized existing in this world. And we can find the best one. And then we got very short list of compounds, which may be 10 or 20 compounds. Then we do those experimental resources on those good compounds. So that's basically the idea behind this computer-aided drug design. Okay, so the first technique, well, is we have some the protein structure for the drug target. So ideally, we have some structure from crystallographer, from the MR, but in some case, we don't have those structure. we can build homology model. You may heard the AlphaFold, which basically can do the homology model given just a sequence. Okay, so with those sequence, we can build up the structure, and with the structure, we can try to understanding the active site. Okay, so with those information, then we can use the technique to dark those potential compound in the active site and the scoring, and then just pick top compound based on that scoring. Okay, so with this, we can screening millions compound. In my lab, within 4 hours, we can screening the 70 million compound. Okay, and then we got top compound. That would be folks for pass to the experimental group for testing. Okay, so this is a very simple technique, but actually it's revolutionized the drug discovery. Okay, many successful example. Okay, so besides that simple tool, we have the physics to help us to set up some the physics-based model we can working for all protein. Don't have depend on the database. Okay, so for this, I want to remind you, okay, to see the simulating the dynamic of the biologic system, well, the principle is very simple. Just look like this transformer. Okay, it can change the shape and for its function. Okay, so now it's a car shape. It can run away, and when the robot is a fighting machine. Okay, so protein and biomolecule does the same things. And then for that motion, we know from the high school physics, underlying is the interaction, is a force. Okay, so if we're able to model the force, then we're able to model the motion. Okay, to the biomolecule molecular world, we need molecular level of force, such as covalent bond, bond angle, dihedral angle, you learn in the chemistry, in the organic chemistry. Okay, we also need some the long range interaction, like the electrostatic interaction, like the van der Waals interaction. So if we can came up set of parameters to model the interaction between the atom for the biomolecule, then we can simulate the motion of the biomolecule, like what I shown here, literally is a folding a small helical peptide. Okay, so this give us the power for modeling the biomolecule. And we know for this motion, we thank the Newton. Newton developed its famous equation to simulate the dynamics. So we can use Newton's equation to guide this dynamics, and then we depend on the computing power to apply that law and to guard dynamic we want for understanding the function of the biomolecule. Okay, so give a little bit of history perspective of this field, molecular dynamics simulation. So it's actually start from the 73. Wow, that's almost 40 years ago. So Dr. McCammon basically used the Apple computer to simulate it, not even call the real kind of particle, it's just some particle, okay? And then 600 particle, and he used Newton's laws, very simple kind of force to simulate it 9 picosecond in the gas phase. Well, that's actually start a new field, okay? And then roughly 20 years ago. Okay, so my PhD advisor, Dr. Dawn, was fortunate to use the supercomputer like Cray, and he was able to simulate the small protein and in the solvated environment, and for microsecond, which are millionfold longer than the first study. Well, he told me result actually not good, which you can see from this comparison. Okay, so experimental structure is the red color, and then the blue color is the folded structure. You see, it's not good. But actually, this study has been cited over thousands. It's a phenomenal study to suggesting human able to model the protein structure and to learn from the protein folding. Okay, so then this work inspired Dr. Xiao, who is the computer science professor from the Columbia University, who also the billionaire by writing the software for Wall Street Journal. When he saw the Dr. Duan study, he inspired, why not build a specialized chip to speed up the calculation so we can do larger system with the longer time scale so that we can tackle the fundamental drug discovery. Okay, so he did that. And this is a specialized computer he designed called the Anton. And with Anton machine, that's literally 15 years, 12 years ago. And you see the result, amazing result. With this machine, he able to fold small protein into the native structure and with very good, and this reach the accuracy of the NMR. Okay, and then this study was picked as a top 10 breakthrough by Science Magazine and 2010. Okay, so not one had the fortune to use this Anton computer. Well, we can use a graphic card. So use a graphic card, you can see we can simulate it reasonable system and to got very good results. So I always tell my student, well, with the computing power, you just sleep, do nothing. You wait, and it still follows Moore's law. Every year, it's double speed and half price. You wait for 10 years, you too will become thousandfold powerful. So I will predict in 2030, not far from now, 8 years, you probably can do those simulations just a second, a minute. You can got all your results and you can analyze to understand the insight. Okay, so let's say some of the application. This is I did roughly 10 years ago when I was postdoc. And I want to see with the simulation, can I fold a small protein with a decent second structure, helices, beta helping. And this is what I did. And you can see the protein are collapse and start the folding process. The helices tend to form earlier because it has short range interaction. And then the helping part is harder to fold. So it needs more time to search correct interaction. Okay, so now it's found that stable one. And then for the hydrophilic part, it's packing inside and make stable. Okay, so now it's folded. Okay, for this calculation, actually it took months to finish this calculation. But now that I tell you, this just take hours. Okay, don't need months. And this is another study I challenge. And this drug, especially first-line cancer drug for leukemia, still true for now. And it works by intercalated between the base pair of nucleic acid, basic DNA, and its suggestion is lock the DNA because we know the DNA have to separate so we can read out genetic information. Okay, so Christopher solved this structure. I was wondering how this drug can go into that site. And Christopher suggesting very simple mechanism. That basically means it's open the mouth, you let drug in, and then lock the drug in that place. And I want to say, well, we have simulation. We can simulate it to see whether all this is true. So again, I started simulation 10 years ago. I just put a drug in my system and see how it go. Can it go inside that place? And how to go inside? Does that really simple open mouth let insert? And let's see how it behavior. Base pair open, make space for drug enter. Okay, so now drug can enter. And once enter, now base pairing. wow, this is different from the Christopher's proposal. It's not open insertion, and we define this a flip insertion mechanism. Okay, so and we are excited about this result. We want to send to the JACS, Best Chemical Journal, immediately reject it. And the review asks, can you provide some the evidence to supporting this mechanism? I say sorry, and we just send to a low impact journal. But now you can check the citation for this paper. This paper is highly cited. I think now it's over 70 or over 60, highly cited paper. So from here, one thing I'm sure that if it's gold, it eventually will be shine. Okay, so let's move to some of the more advanced topic. Okay, we know cancer are incurable, but the research changes that viewpoint. We have the case to show cure for the cancer through a so-called designed receptor for the T cell. And this is what it does. It takes the blood from the cancer patient and who had blood cancer. Okay, and then to isolate it is a T cell. We know T cell is critical for immuno fighting. And then it's engineering a T cell receptor into that patient T cell, and then culture that T cell outside the patient, and then grow to a large quantity and check, make sure this T cell receptor can bind to the tumor biomarker. Okay, once pass those tests, now you inject back and then to treating the patient. And here I show you the one baby. And when he was got this trait, he basically sentenced death. And he did this treatment. And this is now where she is. And literally he's cured. Okay. And then along this line, there are six kind of this treatment has been proven. And this bring the hope for the cancer patient. One day, cancer can be treated as the normal diseases. Okay, so how this work? Well, the secret is that we can engineering the antibody to recognize specific biomarker on the tumor, and then this way the T cell can quickly activate the program to killing the tumor. Okay, so this is the cartoon show it. And my question is that how it work at atomic at protein structure level. Okay, so here's the recent simulation we did, and then we pick a CAR T which are targeting the solid tumor and show promising experimental result. And we try to understand how the designed receptor can recognize the signal and pass that information cross membrane and inside the membrane and activate the program to fighting the tumor. Okay, so here we just simply do two systems. So this is the alone APO system that basically has the receptor alone. And then this one, we put the antigen in the system. So this is the whole system. It's basically mimic the activation, right? Remember this bonding will induce response inside. And then if you from the biochemistry, you know the key for activated is some tyrosine residue, signal residue, and which will be phosphorylated. And when this signal will trigger that phosphorylation reaction. So our question is that how could this receptor sense this biomarker and pass that information cross membrane to the response region? How that happen? We're really curious about that. Okay, so with the simulation, and actually we have to resort the Anton tool because the system for this is had 400,000 atom, and then we have to simulate to the microsecond. If you use the graphic card, this week will take years to finish. But with Anton 2, I literally finished this in days. Okay, so then let's see what we got. So amazingly, you see, without demarker, and so this portion is very flexible. It's just move around, try to search for the antigen, and this part is very fixed. It don't move much, and it's kind of hiding from the kinase, which do the phosphorylation. And however, when you see when this bind, it change. So now this part become fixed, and this part become flexible. Well, this will good because this allows the kinase to come in, bind this side, and do phosphorylation. Well, from this simulation, we learn how the signal was transduced cross membrane, and we call this a bonding induced flexibility switch. Okay, so can we got really support? Well, here is a little bit analysis to show that idea. So I just plot center of this antibody domain, and then center of this response domain, which are red color or blue color. And then here I including the antigen domain, which are shown in the black color. Well, you can see clear difference. Before it's bonding, this portion is looking for antigen. Okay, and this portion is hiding for the kinase. And when you bind with this one, now this part activate star for search the kinase. So that's the bonding induced domain flexibility switch. Okay, so hopefully by those kind of study, we can design more potent T cell receptor for treating the solid tumor, which so far no single FDA approved therapy for solid tumor. Okay, so give you some idea what we've done in our lab. So we use those computational technique to develop the anti-COVID drug. So the project I will talk you in couple slides, which are the anti-COVID antiviral drug development. Okay, and then the anti-cancer drug, I just discussed you the T cell receptor design, and then some very specific very kind of the new novel target of the some the second structure in the chromosome, and might be a new kind of class of drug for treating the cancer by lock the DNA, which are blueprint for the organism. Okay, and then we also use the computation to study the G protein coupled receptor, which are the number one class of the drug target for medicine. And in fact, 40% of the current medicine are targeting on the G protein coupled receptor. And here I show you the simulation result. And this is the signal molecule, which when it's activated, it should be leaving this G protein, which is inside of the membrane. And in simulation, we can see that if we bonding the agonists, which are the activate, and will induce leaving of this molecule, activates this protein. Okay, and then we use the computation approach to get active confirmation of the different type of the ligand. And we know for this receptor is very smart. It's based on the different signal molecules do different response. If you have the agonist is activate, it's activated. If you have the antagonist, which are the deactivate, it do nothing. If you have your partial agonist, then it's partially activated. Well, the same sequence, same protein, how to explain their different function? Well, you will argue they adopt different conformation. Unfortunately, Christopher was not able to solving those individual conformation. But simulation can generate those active conformation for different type of ligand. So here I show you, we use the simulation generate different conformation for different type of ligand. So now we got the active conformation. Then we use this structure-based virtual screening identify the compound. And we pass compound to our experimental collaborator. They bought 10 compound. I got feedback. They got 6 active compound. So now we continue the program to push forward. Okay, so that's kind of some of the example in my lab. Okay, so now let's look at a specific example and give you a little bit more detail how we can carry out the compute algorithm to do the virtual screening, identify the top compound. So this study has recently published in the scientific report. So, and you can Google, you can find more detail. So I give you more something something not actually mentioned in the paper. Okay, so let's first start a little bit of background. Okay, so we know SARS-CoV-2 basically is the RNA virus. So its genetic information is coding in the genome is RNA, and for the host like human, don't have the machinery for replicate its genome. So it itself has a gene coding a protein called RNA dependent RNA polymerase. Literally, its main task is to replicate its genome and also replicate its gene. Okay, so the first drug had approved by the FDA to treating the SARS-CoV-2 is remdesivir. Maybe you heard the name. How it work? Well, it's literally is a nucleotide. It try to falsely incorporate into the chain, but it has some modified, so it's not the normal nucleotide. So it will stop the copy of the viral genome. So this way, it stop the replication of the viral genome, so that leading to shorten the time for the patient. Okay, so that's the how this work. Okay, so this is 2020, right? And at that time, no information about the protein structure. What we have is just this drug, remdesivir. And remdesivir we also know it's a prodrug. And inside the body, it have to convert into active form, which shown here. So then, well, can we do something to find some similar drug? Answer is yes. We can do pharmacophore modeling, and we can use the structure of this active form and to search the compound look like this one. Then we hopefully it can do the same thing when it's bind to the protein target. Okay, so this work is 20. So this is the compound we found and mimic. And I don't know if any experimental group tried it yet. Okay, and but at least we pick some compound, and so here the more kind of supporting data for a potential drug property for those compounds. And again, this can be done in the computer, right? Predict drug property and to pick the best one. Okay, so that's kind of the study. And then 2021, and there are some the first solved structure, and this is that polymerase structure. Okay, and unfortunately, there are no RNA in the active site. Well, we can do homogeneous modeling, right? And we can put those into the site and build a structure to holding active site for the drug remdesivir, and then we can put remdesivir here. So this is the homogeneous model we build, and this is we put remdesivir drug in. Okay, we want to learn more how this drug work, and then use that idea. Can we find some the alternative drug? Okay, so here's the structure we build. So this is the active form remdesivir. We call the RTP. So it's bonding to the here, and then this in the uh, polymerase. And then we also have some control. So ATP definitely can bond this side because it need to be linked to this baby chain. Okay, so then we learn a lot. And with that, then we use some defined for core model to do the virtual screening, and and our final select 3 peak. Okay, and with the 3 peak, we want to verify them in the protein structure model. So we did MD simulation and we verify its stability and we characterize its flexibility, all those things, and to see if that's a good candidate. The answer is yes, just put story. And then we also can do the so-called MMPPSA to calculate the bonding energy, which are important, determine its bonding affinity. And actually we show that it's actually can do better job than the existing ATP or the RTP, those nucleotides. So they look kind of promising, promising. Okay, so actually we've been challenged. We've been challenged, and reviewers say, "I don't believe your homogeneous modeling. How could I believe your structure are good?" Fortunately, at that time, and right before the publish our paper, and there solved crystal structure, and that's one. And then we just superimpose our structure with the experimental determined crystal structure. So here you show how good they are overlap. So reviewer basically say okay to us. So that we publish this paper. So from this example, I will also want to give you the confidence. Use computation. We able to capture some reality. Yes, we need validation from the experimental data. Okay, so then with this structure, now we can do more. Okay, so we can use a virtual screening workflow. So later we have the protein structure, and then we can use more elaborated the molecular docking score, and to screening 7 million compounds. Okay, and within days. Okay, and then we use some the drug property filter and to filter to get 14 lead compound. And then we run molecular dynamic simulation and we calculate the bonding kind of energy, and we do some the clustering, try to identify different molecular scaffold, so we have more chance to be succeed. And then we do a lot of analysis and to use those molecular property, to help us to pick the top compound. Okay, so that's what we did. So this is the final piece compound. Okay, so we pick this compound, and we also validated this compound. And I guess this is the first is validation with the existing compound. And so we have to do a little bit of modeling because in the crystal structure, this form covalent bond. So we have broken that bond to mimic the initial bonding state, and we got very good overlap with experimental structure. Okay, so that give us confidence uh, our structure. Okay, so here stocking score, we pick the top compounds, and we use the zinc database because all those compounds has available commercially. We just simply buy it and test it. So we out of 70 million, we pick the top 1200. Okay, so then we do the molecular dynamic simulation to verify those. And this is the control one. This is the drug remdesivir. You remember? Yeah, it's still very stable and bonding and happily in that active site. Okay, save time. We just move forward. So then we did some routine analysis. So the RMSD basically characterize the conformational change, and we really want to check, see if the conformation change be stable, no longer change. So that basically means those RMSD value are flat. So you kind of can say for the control compound, RTP, ATP, for the four compound we found, they look very similar. So we hopefully they have similar property. And this is just to compare them and the RMSD, RMSF, which characterize the flexibility of the conformation, flexibility part of the protein. Okay, so and then the PBSA bonding free energy, and so then based on those we pick these four compounds. Well, and so here the detail. Oh, okay. So this is the previous slides. At that time, it's in press, and I'm glad now it's published, so you can find, you can check. Okay. So the little bit of conclusion. So conclusion is that with the good information about the drug target, and we can use a very efficient computation approach to screening huge amount of the ligand database and to pick the top compound. Okay, and then for top compound, we're able to afford to do the physics-based model, molecular dynamic simulation, and then to really to look at detailed interaction of the drug with its drug target, and then use that information to pick the best one, and even use those insights to help us to pick the good one. Okay, so then through this kind of virtual screening approach, our end product is a few compounds that can be bought from the company and can be put in the experimental testing. Okay, so then for this set, actually we got small grant from the New Jersey Health Foundation, and which give us the 100K to testing how good those compounds, and yeah, so that's what we're doing. And well, without the Gabby and George, you're not able to hear this talk. So yeah, thank you, Gabby and George, to offer me this great opportunity to share our result with the wide audience. Okay, again, without student, I would not able to. Without student, I would not able to present your result. Without the funding agency, so okay, so the work for present, and it's many did by Dylan, okay, which are excellent student, has very good kind of chemistry intuition and very good at organic chemistry. And then the Philip, and very good in the computational skill, even though, oh, he's double major chemistry and computer science. And then here's my team, okay? And so we cluster in a small room and full of the computer, and we just like to play the game. And but we are very happy, and we feel our work can change the world, can make the world better. So that's kind of motivation for us to work together, particularly in this pandemic, right? And we are fortunate we can keep our program going, and so that's why we're spending so much time on the SARS-CoV-2. And so I think that's all. So I think I'm good at my time. So now I'm more than happy to take your question.
Thank you very much, Chan. I'm sure that everyone here will agree that that was a fantastic and really insightful presentation, and it's a pleasure to have you speak with us. I'm now going to move into the Q&A session. In order to submit a question to our speaker, you should enter your question into the box on the right-hand side of your screen and click send. We will answer as many questions as possible during the time available. And I can see that we've already had some questions submitted, so I'll move straight on to these now. Dr. Wu, your first question is, Why can CADD dramatically cut the developing time and the cost for drug discovery?
Because we don't do the blind testing. Okay, traditional way is just you put the compound in the test tube, and you wish good luck. You not use the information about your target. So then we use a computer. We take advantage of all existing information, the sequence information, the structure information, and then the activity information, and all those data. We can use that. And use those data then can help us to pick the best compound. So that's why you heard of the applied machine learning AI in the drug discovery. Yeah, that's the active field. And you can use existing data set and to build up the model and to make the good prediction, particularly for example for the drug property. So that way you can cut a lot of the time, a lot of cost by focusing on the good compound.
That's perfect. Thank you. Your next question, Dr. Wu, is why is MDS considered a more accurate method to model ligand protein interactions?
Okay, MDS is molecular dynamic simulation. So the molecular docking is a very cheap way, but it's based on the model, so-called the log key model. So that basically means the drug is like the key and the proteins are like log. So they have the shape complementarity so they can match. So this is a very good approximation, but it's not accurately. So reality, when the drug interacting with the protein, it's more like you eat spaghetti. You use a fork to touch your spaghetti, and then you change shape of spaghetti, make the face to your fork so you can eat. So reality is so-called induced fate bonding. And for that induced fate bonding, the docking not able to handle that because docking assumes the rigid body uh, and molecular dynamic simulation, well, it's based on physical model. So all the atoms are flexible based on the interaction with the other atoms. So it's basically can sense the interaction, can change conformation. So molecular dynamic simulation able to change conformation, to capture those conformation change, to really capture the bonding induced conformation change.
Perfect. Thank you for that. That's a great answer. We've got quite a few questions in here, but I'm just going to carry on. Your next question is what is the MMPBSA binding energy?
Okay, so full name is the molecular mechanic Poisson-Boltzmann uh, and the surface area equation. Okay, so this is the physical model to model the solvation effect. And we know the water solvent are critical for biomolecule, but use the X-list water molecule computation is too expensive. And there are some physical models so-called use the dielectric constant, which you may heard from the high school physics. The water has a dielectric constant 80 because it's basically can screening the electrostatic interaction. So then we can use dielectric constant to model the solvation effect. Okay, and that equation is called the Poisson equation. And then with that, we can calculate the solvation effect with a very cheap method. And then the SA is a surface term, which literally is to try to capture the hydrophobic interaction, which are important force in the drug protein bonding, and which are explain the formation of the oil droplet, right? The oil droplet, they will not dissipate in the water, they forming the droplet. The force to hold them together is the hydrophobic interaction. And why it's a sphere? Because that has a minimum surface and that has the packing the maximum volume and given the same surface by the sphere. Okay, so then we can use the surface area to capture the hydrophobic interaction, which are important. So then this MMGBSA literally can allow to calculate out the bonding free energy of the drug with drug protein and give us the accurate prediction of the bonding affinity, which are the critical parameter for the drug development.
Perfect. Thank you for that. We've still got quite a few questions to go through. I just want to remind the audience, if you have a question for our speaker, please submit it in the Q&A tab, and we'll get to them as soon as possible. So your next question, Chun, is what is the major difference between the docking score functions and MMPBSA binding energy?
Oh, okay. So this is basically continuing my line. So docking function literally is considered as empirical kind of the empirical scoring, and literally it's try to mimic bonding free energy. Okay, but it's based on the empirical data. So that basically means you directly measure the bonding constant, and then use that bonding constant to build out some model, and based on the molecular descriptor of the compound. So then when you have the new compound, and then you just uh, use its molecular descriptor, plug in the equation, and then you calculate the bonding affinity, which are the docking score. Okay, so this method is accurate if the compound are very similar to the database build the model. But if you have the novel compound, then this model will fail. So this is called empirical one. And the MMGPSA is called the physics-based first principle model. So it's based on physics. So that is work for all the compounds. You don't need to do the cherry picking. And then it literally try to calculate all the important physics interaction. So the van der Waals interaction, the hydrophobic interaction, the electrostatic interaction, and then also including a little bit entropy, so that it's calculated the full the bonding free energy. So this is particularly good when we have the kind of the novel compounds which don't have the experimental similar compounds has experimental data. So that's the difference between two approach. So which based on the situation, you can pick one and to get some the good results.
Perfect. Thank you, Chun. That's another great answer. I'm going to carry on with the questions if you're happy.
Mhm.
So your next question is, how does binding free energy determine the dissociation equilibrium constant, then IC50 or EC50?
Okay, so this is actually critical. We calculate the docking score, we calculate the bonding free energy, but how this link to the drug, link to the drug property? Well, and thank for the Boltzmann. And Boltzmann basically give us the basic connection between the bonding free energy and equilibrium constant. And the equilibrium constant basically linked to the concentration. And the concentration basically linked to the dosage we use for drug. Everyone wants the small dosage of the drug which have less side effects, and that dosage IC50 is determined by the equilibrium constant, and which are determined by the bonding free energy, which are mimicked by the docking score. So that's kind of logical chain. So that literally, if we have a good docking score, that's good bonding free energy, and good bonding free energy will determine the good bonding equilibrium constant, and the good bonding equilibrium constant literally is indicate very, very low IC50. And IC50 is called the effective concentration for the drug. Okay, and so as so then we can use very low kind of the very low kind of the concentration of the drug, and then it's translated to the dose we use. We use 1 pill per ml or 2 pill per ml, and twice or once per day. So that determines the final dosage. So that's the logical chain. So by improve a little bit our docking score, our bonding free energy, actually we can make a dramatic change. And this again due to the Boltzmann relationship. And the Boltzmann relationship is not linear relationship. It's exponential relationship, which basically means a little bit energy change can dramatically change the concentration. So, for example, a little bit mutation to weakening the bonding free energy by a little bit, it will translate to drug resistance. Now your drug no longer be effective. You have to use a tenfold more concentrated drug to treating the diseases. Reversely, it's also same. You can make the less potent drug to extremely potent by adding a small interaction, a hydrogen bond, then you can make your drug 100-fold more potent. So instead of use 100 peer of the medicine, you just use a single peer. So actually, this is a very critical for the drug discovery to kinetic computation into experimental measurement.
Perfect, thank you. So we still have some more questions in, and I'm eager to get through them in the time that we have available. So your next question is from Afsena, which is, how do you design an agnostic peptide? It is easy for me to design antagonistic peptides. So basically, she's looking for designing
Yeah,
an activator peptide.
yeah. Afghanistan. This is a very excellent question. Yeah, the reason is the structure you use most likely a inactive conformation. Because inactive conformation is a low energy stable, so it's easy to solve in the crystal structure. So then your conformation limited, you found the agonist. Okay, so my solution to this is very simple. You pick the agonist peptide, you dock, put in the receptor, you run molecular dynamics simulation to generate the active conformation, and then use that active conformation to do the virtual screening, to do the docking, peptide docking, to do the virtual screening. And then if you have more computing resources, you can do the molecular dynamics simulation. So actually, we did our drug discovery, what we found are actually the agonists, not antagonists. Okay, so we did some the dopamine 2 receptor, and we generate the active conformation, and then we targeting the agonist, and our experimental collaborator give us feedback it's agonist. Okay, so I think that's my answer for you.
Thank you, Chan. I hope that answered your question, Afsana. We have another question in, Chan, from Abid, which is RDRP essential for transposon control?
Oh, actually, I don't know the answer because the transposon, I believe in, may not have a directly connection because the RDRP is responsible for replicated the RNA, okay, replicate the RNA gene, and transposon, if transposon is part of that one, so then at least this polymerase not able to sense that one. So my answer is that does not directly able to sense transposon, but it can indirectly sense transposon by the host immune system, because the selection pressure will pick the strain with the genomic which have the good transposon and favor the fitness of the variant. So yeah, actually we have some study on that one. So hopefully next time I can present some study on those genomic and sequence level study to answer your question. Thank you.
Thank you, Chan. That's great. We still have some time for a few more questions. Your next question is from Pearly, which is, how do you decide for how long to run a molecular dynamics simulation? What are some guidelines we can use to decide if we need
Yes,
a long or
this is
short?
actually excellent question. Okay, just like you do the experiment, chemistry experiment, you measure the equilibrium constant. You have to wait certain time until the concentration no longer change. Then you do the measurement. Okay, so for the simulation, you have to do the same thing. You have to check so-called the steady state. And so the quick answer is that you can calculate some property, very simple property. For example, the one I use is the RMSD, root mean square deviation, characterize conformation change, and you monitor that over the time. If you see that converge, then you can stop your simulation. But you still see that keep changing, that basically means not reach steady state. And then relaxation time depends on the property you're interested in. So you have to experimentally find out the property you're interested in, the time scale for that relaxation of that property. Then you make sure your simulation time have to cover that time scale, and then you're able to got to converge, and then you're able to collecting the data from the steady state and analyze. Yeah, this is a very good question.
Perfect. I believe we have time for a few more questions, Chan. So your next question is from Mohan, which is it true that some of the molecules identified by computational methods fail in cell-based assays? So the question is, can we somehow incorporate the cell uptake parameter during in silico screen?
Yes, this is drug property, and uh, binding affinity is just one parameter for drug. And for drug property, we need a lot of properties. Admin, right? Is need absorption, distribution, and metabolism, and the excretion, and also toxicity. So some drug even pull, but then withdraw from the market due to toxicity. Well, that's the place of the AI machine learning can play the role. You can collecting all those data and to learn the pattern. What kind of compound would be good? What compound? So like here you mentioned that drug property, then we can build those models. So during the screening, we just feed the compounds and then to do the screening. Actually, a big part of the virtual screening is to optimize the drug property. Yes, we can do that.
That's fantastic. Thank you. So, Chan, your final question for this session is from Pablo, and it is, could this technology get precise enough to replace the need for experimentation on in vivo models?
No. And this is the uh, induce so many approximation. Okay, so one big approximation is that, well, we know at molecular level should be quantum mechanics, right? So here we use the molecular mechanics, we use Newton's law, so that's approximation. And so we use so many approximations, so you're not able to guarantee the result will be consistent with experiments. And another thing is the complexity of the biology. So biology, we have 3 billion nucleotides for the human genome, coding 20,000 protein. And then we have the trillion cell to make our body. How could we put all those things in the computer? No way. So we only can provide approximation result, and then we do need experimental final check. And so our goal is not replace the experiment. Our goal is improve the efficiency of the experimental approach. Mhm.
That's perfect. Thank you, Jan. That's fantastic. Unfortunately, that is all the time we have for, but it's been an absolute pleasure to have you present with us. We've all really enjoyed our time. A
Okay.
certificate of attendance is available to download from the Handouts tab on the right-hand side of the webinar platform. And please feel free to navigate over to our literature section where you can download the latest resources from the sponsors of today's
Uh-huh.
webinar. The webinar will now be made available on demand. So if you have any friends, family, or colleagues that you think may be interested in this
Mhm.
webinar, please feel free to share the link with them.
Yeah. Can you send me the kind of the I do have many students, many kind of colleagues, they interested in this one. Uh, but do you have some the simple kind of the share link that they can click and then they can start to register and then they can kind of watch the video? I
Of
do
course,
have
Dr. Wu,
I
we'll
do have a
send this
lot of
over to you.
back in China. They are really interesting, and so if you have send me that kind of the uh the share link, then I'm happy to put all and in. Thank you.
Absolutely. We hope that
Okay,
you've
great.
enjoyed this webinar and found it
Yeah.
informative and engaging. To help us improve future events, we kindly request that you take a few moments to complete a short survey after the webinar finishes. Your
Okay.
feedback is valuable to us and will aid in future webinars to better meet your needs and expectations. That just leaves me
Okay.
to say thank you again, Dr. Wu, and we will see you again soon.
Yeah. Thank you. Mhm.